Okay, good morning, everybody. And before we start, a couple of things about myself. So I'm a Laurel from the mid-realm. I cook, I do pottery, and I also brew. Mundanely, however, I have a PhD in microbiology, and I teach microbiology at the college level. So this is very much essentially beginner's guide to techniques based very much on how we handle yeast within a modern context. I will mention some of period techniques, but this is very much more based on modern microbiological techniques about handling yeast, making sure that you have pure culture, and also a little bit about how you can catch a wild yeast and make it be something interesting. So I am going to share my screen. And can my moderator confirm that you can see this?
Yes, ma'am.
Thank you. Okay, so given that I teach modernly, means that every class I do has learning objectives. So by the end of this session, you should be able to recognize the tools necessary to capture, maintain, and propagate yeast, describe the methods used for preparation of sterilized media in the home, be able to isolate and purify wild yeast, and be able to prepare refrigerator and freezer stocks, and also propagate yeast for brewing. So this is really what I want you to take out of this class. So before we start, we've got to say, we got to look at it and try and figure out what is yeast. From a biological standpoint, it's a eukaryotic organism. That means that it's a cell like the cells in your own body. It has a nucleus, it has a complicated biology. It's actually a fungus, but it's single-celled, and so it grows in liquid media quite happily. And it has unique characteristics, and it should be recognized that just like we domesticated sheep and dogs and spinach and lettuce and cabbage, we have domesticated yeast by the way that we have treated it, how we have used it, how we have moved it from one brewing vessel to another, how we have, selected and borrowed yeast from people who made great beer and ignored the yeast that didn't make good beer. We have selected for a wide range of different yeast strains. There are strains specific to Europe, to Asia, for bread making, for beer making, for wine making. Yeast have been selected. Yeast, for us, do the simple job of chewing up sugar and generating alcohol and carbon dioxide. When we're making beer, wine, mead, sake, any fermented beverage, we're keeping the alcohol, and for the most part, except with some fizzy, carbonation of beer being an example, getting rid of most of the carbon dioxide. When we make bread, we're doing the opposite. When we bake off our breads, we're getting rid of the alcohols and we're keeping the air. Yeast are ridiculously common.
I'll come out of this.
Could you mute yourself, please? Thank you. Sorry. It is very easy to find yeast. They are on your food, they're in the air. The bloom on grapes is yeast, so it's not hard to find wild yeast. So this is where it gets really interesting, because I'm going to stop sharing, and we are going to go to my YouTube channel, right, where I actually have videos that I prepared earlier. And skip. Start sharing again. I'm sorry. It takes a second to switch screens. So why are you in the way? question for my moderator. Can you see a YouTube screen? Should be canning jars.
Yes, ma'am.
Thank you. Okay, so I am going to mute this. So this short video is just essentially a walkthrough of the equipment. It's the same information that's in the PowerPoint, but we can actually see them. So, okay. Question for the moderator, can you hear the voiceover or not?
No, we cannot hear the voiceover.
Okay, then I will voice over it. So if you're gonna prepare yeast at home, there's some basic equipment that can help you do it. I'm pretty sure many of you recognize what I've got right here was the Instant Pot or other electronic pressure cooker. You need something that will raise the pressure. You need malt extract. You need a scale. You need agar. You need canning jars, glycerol, and you might need little storage plastic vials if you're going to store yeast for an extended period of time. Additionally, you will need plastic plates for making agar plates. Okay, so that's the first thing we need. So the next thing is you are going to need liquid media and agar that are your growth media, your liquid growth media to allow you to catch yeast or grow yeast, and a solid agar plate is going to allow you to isolate single strains of yeast for your use. And it should be noted that I do have a wild strain of yeast that I use to produce beer. It makes excellent Hefeweizens and Belgians, and that's what it's really good for. So so we're going to go through how we set up agar and liquid media. I do not know why. So here we go. So if you are setting up agar, you follow the instructions on the package. You buy the agar on Amazon. And generally it's so many grams per liter. You will never need a liter of media. I generally do it in the 100 ml range. You put your powder into your very special canning jar, just regular canning jar. You add water, and that's it. That's all you have to do to make agar, solid media, or liquid media. The thing to remember here is at this point, nothing is sterile, right? Just using a regular spoon, non-sterilized containers, we're just weighing out our ingredients. Once you have your media and you are ready to sterilize, this is where the Instant Pot comes in. Because the Instant Pot has pressure, and pressure plus time is what is needed to remove contaminating organisms from your liquid media. In a normal laboratory setting, we aim for 15 pounds per square inch of pressure for 15 minutes. Now, the Instant Pot only reaches 13 pounds, which means the temperature is going to be lower. So if the pressure is lower and the temperature is lower, we need to extend the time. And so when I am doing home sterilization, instead of 15 minutes, I go for 30 minutes. The trick with sterilization, you put your media into your pressure cooker. You want to make sure that the caps are loose because you are going to expose these cans to pressure. And just like any other canning process, you want to be able to allow the air to escape a little bit. I have water MEM is malt extract. So MEA is malt extract agar. ME is malt extract. And then I have glycerol. You need to add water in the bottom underneath your trivet. If you do not have water, you are not going to generate steam. If you don't generate steam, you're not going to generate the temperatures and pressures necessary to sterilize your cans. Once you've done and you've got everything ready to go, see here, I'm finally adding the water here. Apparently I was talking a lot. I don't remember why.
So someone asks in the chat, the water that you're sterilizing, is it distilled water or tap water?
It was actually filtered water. I mean, you could use distilled water, but admittedly, I have an RO system on the sink at home. So I'm generally using reverse osmosis water because that's what I have available. If you just have tap water that's run through a filter, that's fine too. And, you know, once everything's in, close your lid, turn it on. If you have a canning pressure cooker, you can use that because a canning pressure cooker will go to 15 pounds. And you can see here that I'm not doing 15 minutes. I think I set it for 30. And that's it. We're done. Everything's ready to go. Oops. Pause. Escape. The problem is everything's hidden behind the so the menus for the Zoom are hiding my menus for moving around. So apologies for the awkwardness of trying to find the next video. If we follow through on the PowerPoint, if you download the PowerPoint, you should find that the videos just play directly from the PowerPoint, but I've noted that in Zoom that doesn't work. So once you are done with your 15 minutes or 30 minutes, however much pressure, here's the important rule. Do not do immediate pressure release. If you immediately release the pressure, if you do the fast release, what is going to happen is that your agar is going to boil, your media is going to boil in those little jars, and they're going to explode, and it's going to be terrible. So always do the natural pressure release, not the instant pressure release. And lastly, the one thing we want to do, once your agar is sterile and once it is cooled, we can go ahead. Let's make that big. I am so sorry. This is so awkward. The agar now has sterilized, it is dissolved, and it is cool enough that you can hold the jar. That's an important fact. You don't want it so hot that you're burning yourself while touching it. And you also want to make sure, because this is now sterile, we have no contaminating microorganisms in here. The plates that we're going to pour our agar in are also sterile. So what you want to do is wash your hands, wipe down the counter space you're working on with Clorox or chlorine bleach or something like that. And then go ahead and start pouring your plates. And you do this by taking your agar plates, your plastic plates out, taking the lid off your agar, and just generally tipping, making sure that you're covering the plates as quickly as possible after you've got the agar in there. And we do this because, frankly, this is a growth media set up to grow yeast. We have yeast in the air. Molds especially love malt extract. And so in addition to yeasts, if you leave agar plates open, you will have a lot of molds. Okay, I'm going to stop sharing that screen and start sharing the PowerPoint again. Choose that. So, like I said, if you do download this PowerPoint, all of the images, all of the videos are linked to from the PowerPoint. So this is really just like how to prep yourself to do the next thing. So if you want to try and capture yeast, let's say I want to capture yeast that represents my terroir, my environment. You're going to sterilize 100 mils of malt extract media, and in that nice little jam jar, and then you're just going to open it, and you're going to put it somewhere that's likely to have yeast. I've known people who captured yeast by putting them under an apple tree or a fruit tree in a window. You can inoculate your agar, your liquid, with something that's already fermenting. Say you've got some fruit on the counter that's just a little bit past its prime and it's starting to smell a little bit alcoholic. There are yeast there that you can capture. You can drop fruit in there that's got a bloom on it, which is yeast, or you can swab the environment and just, you know, put that in there, just like people swab body parts and culture it. Wait for 3 to 7 days. If after that time period you're starting to see bubbles, there's a very good chance you've caught a yeast. There is no guarantee of what you've caught, but you've caught something. The problem is you've probably also caught mold, bacteria, and goodness knows what else, right? You're guaranteed to have bacteria, molds, and yeast in that environment. So what you need to do now is purify your yeast. And the first thing you want to do is remove the mold off the top, because mold needs air. So mold will be growing on the top of your media, on the liquid, and underneath in the liquid itself will be yeast. And then you get a loop and you prepare what is called a streak plate, which again, I have to now. I wish I had embedded these. It would make so much easier. Stop sharing. Share screen. So a streak plate is a methodology that's used to dilute out organisms. And I have a number of agar plates that I have divided, and I've written on the back, not on the lid, because lids can fall off. I've written the date, what I am culturing, and I've divided it into 3 quadrants, 1, 2, and 3. And then I have a metal loop, and I'm going to sterilize my metal loop by putting it in the flame, allow it to cool, and then pick up some bacteria from my culture. I'm going to then spread it in segment 1, just a little bit. Close my lid, sterilize my loop again by heating it up red hot. That gets rid of any of the yeast or bacteria that we'd already picked up. I let it cool. I'm now going to come back to the plate. I'm just going to cool it on the side. I have no bacteria. And then I'm going to pick up those yeast again that I put on the plate, and I'm going to spread them out by moving back and forth. This spreads the yeast out and dilutes them. I'm now flaming my loop again, making it red hot, letting it cool, checking to make sure it's cool by putting it on a bit of the plate that has no bacteria on it or yeast. And then spreading some more in a quadrant 2. I'm going to repeat that a 3rd time. What this allows us to do is dilute out the yeast so that every single yeast cell or bacterial cell in that culture is now going to grow on the plate as a single point of growth. So if we go back to our PowerPoint, which means I have to stop sharing. Where's my share? Did I stop sharing?
Yeah, we don't see your screen anymore.
Damn it. You didn't see the video, did you?
We did see the video. We just don't see that. We don't, right now we just see you.
Okay, that's fine. So, I hate this. You can tell I don't actually Zoom teach all the time. Generating a streak plate. Okay, here we go. If you prepare a streak plate, if everything goes really well, if you look at the image, you look at the picture on the right-hand side of the slide, you'll see that when you start off, you have lots and lots of bacteria or yeast, and so you have solid lines of growth. When you keep going, you end up with these single colonies. And a single colony means it came from a single yeast. When you're trying to get pure culture, the yeast will always be white or creamy and relatively large on this agar plate. Bacterial colonies tend to be a lot smaller than the yeast colonies because this type of agar is not very good for bacteria. So bacteria don't tend to grow very well on it. So you're going to get more yeast than bacteria on malt extract media. Once you have your purified yeast, then that's when the fun starts, because then you get to try and figure out what it is. And you can start culturing for brewing. Now, to do this, you need to take a single colony and put it into liquid. Now, when we brew, frequently we need a liter of liquid media, but I can't take a single colony and put it into 1 liter. It's too big a jump. So it's best to go from a single colony into 100 mils, and then from there to a liter. Now, if you are a beer brewer, you probably already know how to start a 1 liter yeast starter. But the trick with making sure that you have just pure yeast, just the one you need and wanted to culture, you have to be careful when you are inoculating your small yeast culture. And again, I have another video, so I have stopped sharing. I am now going to screen. Skip. Oh. All of, there we go. Okay, so pause. So here is a streak plate that was prepared, and you can see that I've got these nice single colonies of pure, each one of these represents a single yeast that grew into about 1 million yeast. And I now want to put them in liquid media. So to do this, I'm going to sterilize a loop by heating it in a Bunsen burner, aka my gas stove. Let the loop cool because I don't want to kill my yeast. That is a very bad idea. Hot kills yeast. And I'm going to take my jar of malt extract liquid. I'm going to carefully pick up a single colony of my yeast. The problem with canning jars is sometimes they're tricky to open. I'm going to take the lid off, drop in my colony, close the lid. And this is then going to go on a stir plate for about 24 hours. And that is generally enough to generate enough yeast to go ahead and do something more fun with. So the question is, you're a beer brewer. I keep dropping my. How does your yeast taste? So remember, these are non-domesticated yeasts. There's a good chance whatever yeast you captured is going to be terrible at making beer or wine or your alcohol of choice. They have not been domesticated. most wild yeasts are phenolic off-flavor positive. This means they generate phenols, which in a yeast or beer tend to be viewed as clovey, spicy. And so these yeasts have a tendency to work better for farmhouse Belgian, Hefeweizen, and Saison styles. There was a wild yeast caught in Philadelphia, it's now called the Philadelphia strain, that as well as producing alcohol, also produces lactic acid when it ferments. And so this means that it is a naturally souring yeast, which is a very interesting combination. I generally recommend that if you catch a yeast, you do a small 1 gallon batch of beer to see what it tastes like. And preferably pick a simple beer recipe that's just malt with a very neutral hop so that you can actually taste your yeast fermented around about 65, which should restrain some of that phenolic flavors. Because what you want to do is figure out what your yeast is doing. You know, is it going to be able to flocculate, as in does it settle out of the beer? We have a domesticated yeast, so they do that. Is it going to be alcohol tolerant? You might end up collecting a wild yeast that is really not alcohol tolerant at all and can only handle maybe 2 or 3% alcohol, at which point it's obviously not going to work for fermentation. So there's a lot to, it gets really interesting when you get into handling wild yeasts, because they really don't come with all of those lovely aspects of domestication. If you want to bake with a yeast you've caught, please recognize that what we've done is we've generated yeast. So this is not a sourdough. It is simply a yeast. And most likely any flavors you get are going to be indistinguishable from a commercial yeast. You essentially just use the yeast slurry to set up a sponge for your bread and then use that to make your bread. It's going to taste like bread. It may be fast, it may be slow, but any aromatics it produces, the vast majority are probably going to bake off during your loaf. I remember seeing someone a few years ago decided to do a test of, different beer yeasts and wine yeasts for bread making and to see if they could taste any difference between them, and the answer was no. If you are like me and I brew generally at least monthly, I go through a lot of yeast, and I really get annoyed having to spend $5-10 each time for yeast, so I tend to produce my own stocks. You can store the agar plates in the fridge, and those are good for a couple of months, after which your viability of your yeast drops and you really need to reculture again in order to consider them good. You can put yeast in just sterile water. Yeast is very good at surviving under just watery conditions, and that will stay viable for about 6 months, give or take. If you put it in 50% glycerol in the freezer, it should be good for years. And you can then just take some out, streak it on a plate, and do the whole thing over again every time you want to brew. Okay, I have some additional resources here, including a link to the this PowerPoint, which is on my Google Drive, along with other classes. And of course, you can always go to Bootleg Biology. That's the essentially Yeast Wrangling 101, which is a very simple class and took me a whole 30 minutes to do. So I'm going to stop sharing, and we could talk about some other things at this point. So I'm back.
Can we please have the link to the handout in
sure.
the
Give me 1 second. There we go. There we go. I would like to say some interesting things about domestication. that have had really interesting effects on beer. So if one of the big differences in beers is whether the beer is a top fermenting ale or a bottom fermenting ale. And the reason one of those differences occurred was the way that the British versus the Germans propagated their yeast. So the British would skim the yeast off the top and use that to inoculate the subsequent batches of beer. Whereas the Germans would wait for the yeast to settle out from the bottom. They would harvest from the bottom of their vats and use that to ferment the second batch of beer. If we get into the Kvx strains, which are very unique, they were selected to be able to withstand high fermentation temperatures, and the yeast was kept by essentially putting a yeast ring, which was a wooden ring, in a fermenting beer and then hanging it up to dry. So the yeast are incredibly tolerant of desiccation and high heat. So it's a kind of really interesting when you start looking at the genetic aspects of domestication in yeast. We have selected against those phenolic flavors, that pininess, those clovey flavors, and we've selected for things like flocculation, settling out of beer very quickly so your drink is clear, or fermenting at certain temperatures. You know, lager yeast ferment at cool temperatures, ale yeast ferment at warmer temperatures, Kvx ferment at ridiculously high temperatures. So I'm kind of done. We've got like 20 minutes left. So any questions on beer, brewing, yeast, or anything? Hit me.
I'm going to go ahead and stop the recording for the question.
Okay, go ahead.